All right, welcome everyone to this lecture. So different person up here, I'm Michelle, I'm your TA. I think I've spoken to most of you at some point over the semester. I am a fourth year PhD student at the Department of Molecular Genetics, which is the St. George campus, but born and raised in Scarborough, so I'm pretty familiar with the campus and the area. So you may be wondering why I'm teaching the class today. So first of all, I'm very interested in teaching. This is one of three the classes that I TA throughout the year. And so this is a really good opportunity for me to teach in an also different style. I'm used to more tutorials or one-on-one. So a 2-hour lecture is a different style for me to keep learning. And so very open to any feedback that anyone has. And you guys are kind of the expert at lectures at this point. So open to that. Another reason I'm teaching particularly this class is because of some of my expertise in my PhD. So I don't personally use GWAS in my PhD, but a lot of the concepts that I do use are sort of the building blocks needed to understand GWAS. So some of these include linkage disequilibrium and recombination. These are two really important aspects of my work and two very important aspects of GWAS, which we'll learn today. Population genetics and population structure. So I look at a lot of differences in different human populations with different ancestries and environments. And then also just in general, I look at sort of molecular medicine of human genetics. That's sort of the broad range that I'm classified in my department. And so although GWAS can be used on so many different species, and I'm just studying humans, but I'm sort of very familiar with identifying some sort of genetic variant and how it's linked to some sort of phenotype like a disease. And that can be applied across many different species. So with that being said, let's get started with our learning objectives for today. So our first learning objective is to define what GWAS is and to identify why it's used. The second is to explain haplotypes and extend the use of linkage disequilibrium in GWAS. The third is to assess the validity and discuss various criticisms of GWAS. And then the fourth is to analyze the results of GWAS data and identify its breadth of applications. So let's get started with our first of what is GWAS. So the name is Genome-wide Association Study. So let's kind of break it down to understand what it is. So genome-wide, so quite literally all over the genome, and association, some sort of correlation with different variants along the genome to some sort of disease or trait or phenotype. So we're trying to look across the whole genome, identify all the markers that might be associated to a certain phenotype. That's the general definition of GWAS. So there are two kind of main categories of defining a trait. You can define it as a monogenic trait. Mono is one, therefore it is a trait controlled by a single gene. So an example of that is cystic fibrosis. It's controlled by gene CFTR. And then you can have a polygenic trait. Poly means many. Therefore, these are traits that are controlled by many genes. So a big example in this is type 1 diabetes. And so it's a very, very complex disease. And these are sort of the key traits that GWAS works really well on because we're trying to identify as many variants that we can. Positions of our genome that were known to associate with type 1 diabetes. And then once GWAS came about, they were able to identify 64 reported positions on the genome that were associated for type 1 diabetes. So this is a very classic case of the importance of GWAS. Without being able to survey the entire genome, we were only identifying a few positions that were in the association, and so opening it up to the entire genome, they really expanded it from 6 different positions to 64. So these are the type of traits that GWAS works well on, these polygenic traits, those that have many, many genes involved in it, very complex. Okay, so that's our first learning objective. Any questions moving on? So we all understand the very core aspects of GWAS and what kind of traits they're used. Any questions? All right, let's move on to our second learning objective. So this is getting a little bit more detailed now. So explain haplotypes and extend the use of linkage disequilibrium in GWAS. So first of all, what type of variants are we looking at in GWAS? So the common types of variants are our SNPs, single nucleotide polymorphisms, which you've talked about in your class already. So this is where we have a single base pair change at a certain position in our genome. And so this is an example here. We have this small population. Majority of the individuals have this G allele, and 2 of them have an A allele. So this is a single nucleotide polymorphism. There's one change. The remaining, both sides of those have not changed. It's just that single position. So this is a SNP. And this describes a quite common genetic variation. SNPs are usually quite common in the genome, about at least 5% of individuals in the population in order to be called a SNP. And so an important aspect, especially when we talk about population genetics with SNPs, is defining which allele is the minor allele and which is the major allele. So like it sounds, the minor allele is just the allele that is found in fewer individuals, and the major would be found in the majority of the population. And this is important because just based on the sole number of individuals with one allele or the other, we don't actually know which allele came first or which is a maybe disease-causing allele. We just know that there's 2, and we know that one's at fewer frequency than the other. And so sometimes you'll see it as referred to as a reference versus alternate allele. Reference being that it properly matched to the reference genome, as you guys have done your mapping assignments and for the midterm. So you know that it matches to the reference genome. They call it the reference allele. But in that case, you don't necessarily know that the reference allele is the one that's most common in the population. Often is, not always. And so that's why sometimes we use also the terms minor versus major, which is just referring to the frequency of the 2 alleles in the population. And I just wanted to point out that GWAS can be used on other types of variants, including insertions and deletions. This is sort of a really big field, so kind of anything that you can think about testing on, it's probably been done. Today we're focusing on SNPs. This is sort of the foundation to GWAS, but it has been done and it can be done on many different types of variants. Okay, so when we look genome-wide, that is a huge area, right? There's a lot of our genome. And so in humans, for example, we have millions of SNPs. And so how can we realistically test millions of SNPs in a decent amount of time and using enough manpower, compute power, etc.? And so the reason they can do this with GWAS is because of using haplotypes. So haplotypes is a group of alleles that are inherited together on a chromosome. So I always like to use just an example because sometimes it's a little bit complicated. So if we have 5 individuals in our population, let's say here, and I've highlighted 3 sites that have a SNP. So now we have to look through each individual and see if there's like a matching, identical match to another individual. So in this case, individual 1 and an individual 3 have exact matches. They both have A, T, C, C, G, T. That means that is one haplotype. We do to the next individual, that matches with individual 5. So that's a second haplotype. And finally, individual 4 doesn't match with anyone. That's a third haplotype. So even though we have 5 individuals here, we only have 3 haplotypes. So that's exact matches of those groups of alleles. And so in about 2003, there was this international HapMap project, which basically started to map these haplotypes along the genome and allowed us to look at this genetic variation and relate it to different phenotypes, etc. And so basically what it does is, instead of taking every single SNP, as we mentioned, it identifies these haplotypes. So we take these large regions and make it into sort of a smaller subset that we can look at our haplotypes. And then even from there, they find a select few SNPs that actually explains the haplotype combinations. And those are called tag SNPs. So by looking at just these three tag SNPs, we can identify different haplotypes, and then we can go back and identify the SNPs. So this is the key that GWAS uses to go from millions and millions of SNPs in the genome to about 500,000 tag SNPs. And so if we look at our tag SNPs, then we kind of are able to identify genome-wide. So why does this work? The reason this works is because of recombination and linkage disequilibrium. So as we know, during meiosis, we have our shuffling of chromosomes. This is recombination, the crossover of our two chromosomes. And so if you think of our haplotypes again, remember we have the exact sequence. And so let's say A, B, C, the capitals here is one haplotype. Little a, little b, little c is another haplotype. When they cross over, we end up getting 4 haplotypes. So we went from 2 to 4 because now we still have a haplotype of all the big letters, still have a haplotype of all the little letters, but now we also have these 2 recombinant haplotypes, big A, big B, little c. And so this is increasing our haplotypes, which increases our genetic variation, etc. And so this is important for our GWAS and tag SNPs, is because when you have 2 genes that are very close together on a chromosome, the chance of a recombination event happening between those 2 genes is very small. So they are termed to be linked. And therefore the chance of the shuffling of these alleles and new haplotypes is very small. So basically whatever happens to gene 1, you expect to happen to gene 2. It's going to be correlated to each other. Whereas if you have the 2 genes are on completely different chromosomes, they're not linked. And if they're far apart on the same chromosome, they're more likely to be not linked. There's a chance they could be linked, but that far away, the chance of a recombination event happening in between those genes is very large. So that's where you're going to have the shuffling, more haplotypes created, and this is where we get into the linkage disequilibrium. Just a quick note that it's actually pretty complicated as well, even within our genome. There are certain regions of our genome that just have high amounts of recombination. So this was a paper actually published from my lab in 2015, and this is just a picture where they showed a segment of chromosome 1. And so they show, and this is the recombination rate. So the higher the bar, the higher the recombination. And so they show these segments of the genome that have really, really low regions of recombination, even for a span of the chromosome, and then some other regions which have these like giant peaks of recombination. So rule of thumb is what I mentioned here. If they're close together, they're very likely to be linked, and if they're far apart, they're likely to be not linked. But even within those regions, it does get a little complicated as well, which is what I study, so I find it interesting. Okay, so yeah.
So you're telling us that there are certain regions that have high recombination rates, but like, why do those certain regions have it? And is it like just on where it is on the chromosome, or is it because of like particular traits that need to be developed, for example?
So this is based on.
Repeat the question.
Oh yes. So the question was, why do certain regions of the genome have higher rates of recombination? Is it based on where it's located or other factors? And so there is known to be like one particular gene that responds to, like, if it's found in that region, it's a high recombination region, and so it's based on sort of the recognition of that it's a zinc finger protein, and so that it causes these sort of higher rates of recombination in certain regions. So it's not a very straightforward answer, but it is quite known which regions, we have a map of, at least for humans, and I think for others as well, we have the map, so it's pretty known which regions have higher rates versus lower rates. Yeah.
Are centromeres considered cold spots?
Are centromeres considered cold spots?
I think there's no combination happens there.
Yeah, I'm not too sure that
Yeah, I don't. There's.
That yeah.
Just hold it together. Just hold the 2 strings.
Yeah, it's super cold.
Yeah, so it is referred to. So the centromeres are referred to cold spots because there's no recombination. Any other questions here? Yeah.
Does everybody use the same tag SNPs, or everybody uses different tag SNPs?
So the question is, does everyone use the same tag SNPs, or will they be different? They will likely be different tag SNPs. They're going to be ones that you sort of define from your genotyping, and then it will be whether they're sort of linked to other SNPs. And so we'll go over sort of what it means to be associated to tag SNPs, but yeah, the tag itself it's more just defining a region as opposed to very specific SNP. Any other questions? Okay. So back to our linkage disequilibrium. So another way we can think of it is looking at the difference between linkage disequilibrium versus linkage equilibrium. So I like to think of linkage equilibrium as if every single position in the genome was independent. So if I'm a position, there was a recombination event on my right side and there was a recombination event on my left side. I'm not linked to anything. It's completely independent. And so this is sort of this image at the bottom here where you see all those lines. It's showing that those are all independent positions. So that's equilibrium. That's not usually the case. There's a lot more going on. So usually we see linkage disequilibrium. That's where we have these larger bars. So that means that everything in this bar are correlated to each other. They're all linked. So that's linkage disequilibrium. They're not independent positions. And so the reason that this works for GWAS is because let's say we take this segment that are all linked together and we have our genotyped SNP here. So if we test our genotyped SNP, we don't know that there's a disease risk SNP here, right? That's what we're looking for. But if we have a genotyped SNP at this position and we test it on our GWAS, because they're correlated together, we will identify that this entire region has some sort of SNP that is correlated with the disease. And so that's why GWAS works so well, is because of these large blocks of linkage, we don't have to test every single SNP. We just test one, and it'll tell us information, including if there is a disease SNP in that block. And so this image is just sort of another way to look at it. So we can, again, we have a disease locus in red here. We have a marker locus labeled as number 2. So this marker is the one that's a tag SNP that we're looking at in GWAS. And in the majority of the population, which is shown in these ones, highlighted in yellow is those that are linked. Most of them have linkage between those two sites. And so again, if we use our marker locus to test for an association, we are going to pick up an association because it's linked to this disease locus. And so based on that, when we have a significant association, that can be one of two things. It could be that we found the disease locus, the position that we genotyped and the position that we had our tag SNP is in fact the causal position that's causing or including in the causal relationship to this disease. Or it could just be one that's in high linkage with the actual disease linked. So we could have, maybe we chose number 2 as our genotype. It's still interesting to see that there's an association, but it might not be direct. Yeah?
Could you just explain the picture in the last slide? Like, why is there like 1s and 2s and why like the yellow region is different?
Yeah, absolutely. So the 1s and 2s are just, so this is sort of like the marker position, like the tag SNP. And so those would just be different alleles. So if it matches with the 2, that means that all the 2s have the same allele, and all the 2 1s have the same allele. And
that's the same as like the first slides where it's like all G's and then there's an A.
Right. So the question is it the same as the first slide? So yeah. So that's identifying one allele versus the other. And then the yellow is showing that block of linkage disequilibrium LD. And so because there's no linkage, that's why we see a 1 instead of a 2 here. Whereas all the places where we have linkage in the yellow, we see the disease and the number 2 in every case.
Okay, thank you.
Any other questions? Okay. And we understand this part as well. So if we have a significant association, it's talking about the whole region. We don't quite know at this point whether we have the locus or just one that is linked to the locus. That makes sense? We're good with that?
I have a question.
Yeah.
So if a person has the 1 that's not linked, that they don't have the disease, is that what that means?
Not necessarily that they don't have the disease, because they could still have the disease locus, but it's about sort of the significant, like we're going to have a significant test. And so if the majority of those that have the number 2 also have the disease, then it would count as significant. There can be a few that that's not the case for, but it'll still be statistically significant. And so these individuals likely still have the disease because they have this SNP in the disease locus, but the tag SNP would show as if it was sort of an abnormal, like they don't have the SNP in this position, but they have the disease. But based on statistics, it would still pick up the association. Okay, and any questions online? I'm not seeing any right now. Any?
Nothing.
All right. Okay, so let's go through sort of a step-by-step of what we're looking at here. So let's take sort of a simple example of height. So we have obviously a variability in height. And so this is a continuous score. GWAS can be used for a more discrete score, just like yes, there is a disease, no, there isn't. This example, it's a continuous score, so everyone has a sort of value associated to their height. And so for every single candidate variant that we have, we're going to look for this association, this sort of pattern. So we'll have the phenotype on the y-axis, in this case it's height, and we'll have the 3 genotypes on our x-axis. So we have our heterozygote in the middle and the 2 homozygotes on either side. And so if we see something like this, where there's a linear association within those genotypes, and we have a p-value that shows it's significant, this would be a significant association. So this means that the tag SNP that we are testing is associated, or at least the region that it's linked to is associated to this phenotype, which is height. And so then this is just a reminder, so the test in this one was this 1, 2, 3. It might not be the actual causal variant, it could be the one that's linked to it, but we're at least identifying the region that is of interest in terms of this association. So this would be a linear model that we're testing. And so this is done on every single tag SNP separately. So you're not going to look at the figure of it for everyone, but there's an automatic test that you can do to test for these associations and collect the p-values. And so once we have the p-values for every single SNP that we're looking at across the genome, we look at a plot like this. This is called a Manhattan plot. So we have our chromosome position on the x-axis, and then on the y-axis, we're looking at the minus log 10 p. So this is the p-value. We're taking the log of it because when you're testing, when you're saying something is significant, that's usually below a threshold of, let's say, 0.05. So it's a really, really small area, right? So we take the log to sort of expand that so we can actually see differences within the p-values. Which ones, although they're both significant, which is more significant than another. So that's why we take the log. And then we put a minus sign just because the way our brains work, it's usually if something is higher on a plot, it's greater than, more significant. So that's why we put the minus, so that anything above, anything high on the plot is the most significant. And then they often put sort of a dotted line to actually show the significance threshold. So in this made up example of height, we have 3 positions that have shown a significant association with height. And you'll notice we get these like long sort of peaks here. So this is showing us, we know that there's linkage, and that's what the linkage is. But we'll take the very top SNP in that peak because that's the one that has the smallest p-value, the most significant. And so in this example, they identify, or it's a made up example, but there's 3 based on those peaks. Yeah?
Do you feel comfortable explaining briefly what a p-value is for those who don't remember?
Sorry, can you say that again?
Remind what a p-value is for those who don't remember.
Oh, p-value, yeah. So this is describing, so let's say our p-value is 0.05. So that's saying that there's a 5% chance that the value got is as extreme or more extreme than if the null hypothesis, which is nothing's interesting is happening, is true. So if we say that, so there is a chance that, there's a 5% chance that it could just be something that's happening purely by chance, but because it's such a small percentage, it's likely to be a sort of biological significance.
So on the slide, it only shows like the height versus like where did RS456 go? Like it's not in the 2 or 4.
Okay, so the question is, where is RS456? So this was, the tag SNP is RS123, so that was one that's tested, and that's the one that's going to be associated. This number 3 is just showing that it might actually be 456 that is the causal variant, but we didn't test that. But because it's linked to 123, we're still picking up that association with 123. So it's just sort of the idea of this tag SNP again. 123 is the tag, and it's picking up an association even though anything linked to it could be the actual associated or causal SNP.
Well, that's the same with RS370 and 789.
So this is a different position. So the question, was, is this the same for RS370 and RS789? So these would have their own plot. So this is just an example of one, but this would be done for every single position. So they would have their own group of box plots and p-values associated to that position. Yeah.
Okay, so just for clarifying, I'm not entirely sure on the linkage disequilibrium. So say if you were to apply that in this context, and say if you can apply, how would you explain it in the context of linkage disequilibrium in this example?
In this example, okay, so the question was a little confused with the linkage disequilibrium and how can we sort of explain it better using this example. So in this example, we got RS123 as a tag SNP, right? We're just testing to see if it's associated with height. And so in this, based on our linear models, we see, yes, there is a significant association with this and height. But what we don't know is it actually this tag SNP 123, or is it something nearby this? That's where the linkage comes in. So let's say in reality, it's RS456 that is the actual causal variant. We didn't test directly whether 456 is associated with height, but we did test if its linked neighbor 123 is associated, and we say yes, it is. So it's sort of more identifying the region as opposed to the very specific SNP, because we can't test every single position. So that's why the linkage works, because it's, if RS123 is significant, then it's describing all the regions linked to it.
So there will be multiple genes affecting height. That makes sense. Like if we're describing it as a region, if we're not sure exactly what, we can't test exact, like with the exact atom. We would assume the region, so it would be polygenic. All right.
So the question is it going to then be many genes? So if the SNPs are close by to each other, there's a chance that they're still in the same gene. But it's more just describing the region, and then there can be further studies, which is actually, I think, the next slide. So there has to be sort of a next step after GWAS to actually identify, okay, which one in the region is actually the one that's the causal variant. And so you're just identifying the region of interest, essentially. Yeah.
I just want to make sure of something. So we're doing the box plot for each tag, right?
So the question is if we do the box plot for each tag SNP. It's theoretically yes. If you're running the GWAS, you're not going to make it for every single one. This is what happens sort of behind the scenes. The box plot is just a good visual example of what we're looking for a linear model. In reality, you just sort of you run your code as you guys have been learning, and you'll run many, many linear models, and then you'll just sort of get a list of those that are significant.
And the Manhattan plot is just summarizing that for each SNP tag. So
Right.
each line in the other plot is like a SNP tag, right?
Right. So the box plot is, you're right, is looking at one SNP at a time, and the Manhattan is summarizing all of the ones that you've tested, all the tag SNPs across the positions. Yeah.
Thank you.
No problem. Yeah.
I just have a quick question. If RS123 wasn't the causal variant, would it be lower on the Manhattan plot, and then the actual, I guess, causal variant would be the highest one?
Not necessarily. So part of it is in this example at least, we didn't actually test 456. So you are sort of making the assumption that, so let's say like one below here is like this one next door to RS123, just as an example. So you do take the one that is the most significant, assuming that it would be more likely to be the causal SNP, but it's not necessarily. So again, you sort of take it as knowing that it's in linkage with something that is causal, and then it's the sort of fine mapping functional work downstream that can actually help to identify which one is definitely the causal. Okay. Any other questions? Oh, and I just put a note here. So this is an example using a continuous, right? So we said height is continuous, so we're using the linear regressions. If you're using a categorical, it would be a chi-square, just sort of for any stats people out there. That's what will be done. Yes.
Just adding to that question. So like, let's say RS123 was like number 1 on the Manhattan graph. Would that mean it's not causal then? Or
So on the x-axis, you mean, or?
Like, RS123 is like on the Manhattan is 6, right? I don't know.
So this is the chromosome position is on the, so that's just showing where, so the RS6 is just on chromosome 6 in a certain position. So this is just showing the position across the genome.
Okay.
So the numbers are not based on the strength of the association, it's like the height of them. So the taller they are up here is where they're significant. So anything like here, these, although are showing some sort of relationship, it's not a significant relationship, so that's why it's below these lines. So we still see those peaks, but they're not significant.
Wait, I just have a question. So
Yeah.
if like something goes above threshold of significance, do we quantify the level of significance, or is it just like a yes or no?
So usually you will see this line. So this is meaning that, like, you know, those that are very close together are all showing a significant association, and so we'll just take the one that's the highest, which has the lowest p-value. That's sort of rule of thumb, and then like I said, you'll do sort of downstream analyses to actually pinpoint specific, but you'll just take the one. That's also why we take the log, is to be able to compare those difference in those really small p-values. Okay. Oh, and I just put a fun example here. So the reason it's called a Manhattan plot is because it looks like the Manhattan skyline. So we'll see these large peaks, just like buildings. So I had put a break here, but we're not quite halfway to 4 o'clock. Do we want to take our break now, or do we want to wait a few more minutes until 3 o'clock? We're tired? Okay, let's take a 5-minute break. So we'll come back in 5 minutes. Okay, welcome back everyone. So let's continue on with our GWAS lecture. So we're moving on to our third learning objective, which is to assess the validity and discuss various criticisms of GWAS. So the first sort of key to GWAS is we need to have some sort of validation. So essentially what this means is when we test across the genome, we often find to pick up a lot of false positives. And so sometimes they refer to this in early days of GWAS as the winner's curse. You know, you pick up significant associations and you got excited because you found some sort of association with this disease and it's really exciting. But in reality, if it's a false positive, you're gonna, publish something that is not actually true. And so instead what you need to do is have a validation, which is essentially completely independent data set. And so whatever conclusions you got from your GWAS, you have to now test those same SNPs on your validation, your independent data set, to see if it's still significant. And if we still see that it's still significant, there is a significant p-value, and also the direction is the same. So remember, we're looking at our linear model of our genotypes. We want to see that same direction of effect. So if both of those are the same, then it is likely that it's not a false positive, then that is actually a true significant association. And so often in earlier days of GWAS, this wasn't necessarily done in a particular paper. It would be done like in a separate validation study. And so this is an example that I got. This was published in 2016. So there was a previous GWAS that identified 2 SNPs with an association with sort of a clinical outcome related to renal transplant patients. And so that original paper has been cited many times, and people are learning from it. And then a study came out in 2016 which actually tests those 2 same exact SNPs on just a separate group of recipients, still based on renal transplant, and they saw no significant association. And so this paper that had published this great result, those were actually false positives. And so nowadays, you will not see a GWAS paper that does not have a validation already included in the original paper. That is just gold standard now. Journals won't publish without it. So you need to have an independent data set testing on a completely different group of individuals to see if you get the same result. Yeah?
How many individuals would you have to test for you to be certain that's an association?
That's a good question. So how many individuals do we need to test to be sure there's an association? So this, I think, is a more complicated answer. There's ways you can actually test to see that. So it would be based on sort of like the significance level. So I don't know if there's like a general, I would say you usually see, I'd say minimum 200 individuals in a study like this, but nowadays you'll see much more than that.
So what if you have one study that shows association and another independent study that shows association and one more, but then it doesn't show association? Then it's like, well.
So the question is, what if you see maybe 2 studies that have an association and a third that doesn't? I would actually think that's interesting, because what's happening, what else is different about those, right? Are they from a different population? Have they gotten different treatments, right? I would think that there'd be sort of a next level analysis that can be done to see why, or is it just by chance it didn't test as significant? But there might be more going on there. But usually if it's found in at least 2 studies, then it's pretty well accepted that there's a significant association. Any other questions with this? Yeah.
So this just out of curiosity. So say if you were to conduct a study regarding this so would it make sense if we hear they're talking about the transplant of was it renal?
Yeah, renal transplant. Yeah.
So what if they tried to do the same study, but like maybe take where this individual might be from? So like if you were to test in a certain area, so like if you're going to do another study with that population, right? So how would you say that would work, add to it? Is there a study that's been done like that?
Yep, that is a great point, and we are going to get to it. So the question is, what if you do a validation in a different population? That is a great validation to be able to test across populations, because especially for, if we're doing something like a treatment or something like that, and you're testing primarily on one population, and you have someone come into your clinic that is from a different ethnicity, and you're just sort of assuming that it's going to work for them too, it might not. So having, validation on a different population, excuse me, is a great validation. And if you don't see differences or you don't see it significant, maybe it is based on the population and not necessarily that it's not significant or it's a false positive. Did that answer your question? That's what you're getting at?
We're not going to get into it.
Okay. Any other questions here on validation? We get why it's very, very important. And this isn't just for GWAS, for all genomic studies, it's really important to have sort of this extra data set to back you up. Yes?
So what do they call validation? Is it finding the causal SNP, or is it making sure that the tag SNP is actually associated statistically?
So the question is, what is the validation looking that the tag SNP is still significant, or we're looking for the causal? In this example, so we're looking at to make sure that the tag SNP is still significant. The causal will be more of the downstream sort of functional work, but it's a completely different type of analysis. This is doing the exact same analysis, just on a different set, same tag SNPs, and to see if the tag is still significant in this new cohort. we're good with validation. So we're going to move on to another important aspect is multiple testing. So I mentioned what the p-value is. So there's a 5%, if we have a p-value of 0.05, there's a 5% chance of seeing the same value or a more extreme value or result if the null hypothesis were true. Null hypothesis being nothing's happening, it's not significant, it's just by chance. And so what this means when we test a lot of SNPs, so example, let's say we test 20 SNPs, that means we're going to have one of those 20 that is significant, which is a false positive. That is the 5% chance of it being extreme but just by chance. So you're going to have 20 SNPs, you're going to have one that is quote unquote significant, but there's not actually a biological reason for it, it's just by chance. Okay, so one's not so bad. But if we're testing 1 million SNPs, that's 50,000 false positives. That's not so good, right? If we have a data set and we pick up 50,000, and you don't know which ones are false positive, right? So you end up having all of this data, you don't know which ones are true versus false positive. That's a problem. So that's big issue with GWAS because we're testing large number of SNPs across the genome. And so this is where multiple testing comes in. It essentially adjusts your p-value based on the number of tests you perform. And so there's a number of ways to do this. The detailed examples I don't think are necessary. Some sort of important ones, there's one called a Bonferroni correction, there's one called a Benjamin-Hochberg correction, and there's one called a permutation test, which is sort of the gold standard of GWAS. But the nitty-gritty of those aren't important, it's just the overall multiple testing, adjusting that p-value based on the number of tests, which in GWAS is large. Do we get that? Understand the importance of multiple testing? So lots of places in GWAS for false positives. So both the validation and the multiple testing are important to sort of try to keep that number of false positives low.
Can you just say that again?
Yep, so multiple testing is to adjust the p-value based on the number of tests we perform. So it's going to lower the number of false positives. And so because for GWAS we're testing large number of SNPs because we're testing across the genome, it's really important that we are reducing the number of false positives, which also the validation cohort does. Otherwise, in this example, we said if we test 1 million SNPs, we'd get 50,000 false positives. We don't want to publish a paper with there's 50,000 significant sites. That's so exciting. This trait is so complex, when that's not true. It's just based on the chance of a test. Good? Okay. Oops. So the next thing I want to talk about was population bias. This sort of addresses the question that I mentioned or answered earlier. So in human populations, at least in GWAS, it is predominantly done on European populations. And so this is there's a GWAS catalog for human GWAS, and this is an image that I took from there. And the ratio is about 10 to 1. 10 studies that are done on European ancestry versus the one which is done on all others combined. So a huge difference. And why is this important? Well, we don't actually know for each individual study whether the knowledge gained can be transferred across these populations if we don't test it, right? So as suggested in the question a few slides back, if we have a validation on a different population, that's good because then we can see, is it transferable? Are we still picking it up when we're looking at a different population? And so an example of this that I thought was really intriguing, so there was this HLA variant that was found to be strongly associated with this syndrome, Stevens-Johnson syndrome, which is based on a severe skin sensitivity reaction to a particular drug. And when they tested a cohort of Chinese patients, they found it in 100% of the patients. But when they tested it on European, they found it in 0%. And so I thought this was just a compelling example because let's say they started off with the Chinese population, they tested it in 100%. Well, maybe they would get rid of the drug because they found 100% have a skin sensitivity to the drug. Well, let's not put it on the market. When in reality, well, maybe it does help a different population, right? Or reverse, if they only test it in European and say, great, no one has a skin sensitization, and they put the drug out, and then all of a sudden people with Chinese ancestry are getting this irritation, they weren't expecting it. So that sort of shows the importance of, we can't necessarily transfer the knowledge we know based on these different populations. And this isn't just for GWAS, this is a really big topic in many genomics or science in general. So I hope that you're exploring this in your other classes as well. Oops. And so this was a picture from Nature Reviews. It only goes up to 2016, but it shows that especially early GWAS was almost 100% European populations, and then started to get a few extra in the mix, but it's still predominantly European. And so what are some of the biological reasons or sort of more technical reasons why? So one of it would be data availability. So we have sort of these big population cohorts, such as the UK Biobank, that's based in the UK, that's one of the biggest population cohorts. So it's going to be what data is available. We also I use a cohort from Canada, again, predominantly European descent, so it's based on what is available. Another point is admixture. So this is, as humans, we are very diverse, even within an individual. And so sometimes that makes things more complicated. And so some research, at least early stage sort of, if they pick up individuals that have sort of a mix of ancestry, they might remove it just to try to keep it as simple as possible, which is great for sort of more the fundamental biology. But as we get to things like that we are trying to transfer into the clinic, you can't assume that you're gonna not have someone walk into your clinic that does have a mix of ancestry. So that's why we also have to test it on these admixture populations as well. And then more biological even from that, the linkage disequilibrium, the LD, is actually reduced in African populations. So what does that mean? So African populations they're older, they're larger, and so they have more genetic diversity, more haplotypes. And so the blocks of linkage are going to be smaller. That means you're going to have to need more tag SNPs to actually test across the genome. So you just need basically larger genotyping rate. It's still possible, but that is one thing with the different population size of Africa as an example. You do need more SNPs being tested because they'll have smaller linkage blocks. Yeah.
I'm just wondering, is this population bias something that you think or would change, like it's changing in the future, or is it something that you think would be more persistent for even longer periods of time?
So that's a great question. So it's whether I think that this population bias will be sort of persistent in time or if it's changing. From my perspective, I think it will change, not right away. So I know that for my in my department, we have a lot of sort of EDI related seminars, and it's a lot of students who are asking, well, what can I do? If I'm just given this data and it happens to be from Europeans, what can I actually do to try to diversify? I'm just a student, right? And so I find that those are usually, at least from my department, the people who are asking those sort of important questions are students. So hopefully as that generation continues on, they take that, and when they do actually have the power to decide what participants are being sampled and things like that, then they'll mix it up. Even now there are more populations being sampled, but also to think that the European samples that we have aren't going to go away, right? So maybe we put a pause on those for a little bit and sample some other populations and use our resources for other populations. So I think it will get better, maybe not in the next short term, but over time it should be. Yeah.
Does this mean that there's a SNP that's causing a disease in the population and might not cause a disease in the other?
Great question. So the question is, does that mean that if there's a SNP that causes a disease in one population, it might not cause it in the other? That is possible. There's a lot of factors, including environment factors that can contribute it. It could also be, it's very complex, especially in these types of polygenic. There's many. Right? So if it's not just based on one SNP, it can be based on the rest of the genome. It can absolutely affect based on these population structures. So it's actually really important if you do have a sample that does have a lot of populations, you can correct for this population substructures as well. Yeah.
So adding on to her question, so with sickle cell anemia and I think it was malaria, I'm not entirely sure, but with those, would that be like a good example for this question?
Yes, that would be a great example. So that was the sickle cell anemia with malaria example, which
It's just homozygous and heterozygous, so one SNP doesn't affect another population while this does affect.
yes, exactly. Where I'm trying to think of the exact example with the sickle cell, but it was basically where it affects, it is, what was it, protective of sickle cell in those countries that have malaria, because it sort of has this resistance that, right? Yeah, so that's a great example. Whereas if other countries aren't exposed to malaria, they don't have that same like genetic variants, then they are more prone to sickle cell. That's sort of the classic example there.
Also, why is it mostly, why are we mostly looking at European samples? Is it just because of the availability?
The main reason is data availability, yeah. The big population cohorts that we have is from Europe. And yeah, so it is improving, but that it's just based on what we have available. It takes a long time to sequence and to do all this, and that's sort of just, those studies started earlier, essentially, so that there's more of it. And so, and you can reuse it, right? So if the UK Biobank, I guess, an example, is a really big population cohort, it doesn't go away. People are still actively using that cohort and that set of individuals for a different question, and there's tons of phenotypes to test and things like that. So that's why it's primarily on Europeans. Yeah.
I have a question, I guess more like your opinion. How important is it for us to tell people who aren't of European descent when we're publishing these studies that this might not apply to you, I guess, because you can't read that in a paper. Like, it seems like, oh, it's so generalizable. I can take this drug, whatever it is, like on the clinical side. But how is it important that this information is disclosed? Not just like to be in a lecture, but like to the people who are patients or people in the clinical setting.
Yeah, that's a great question. So how important is it that this is actually disclosed? And nowadays, that's gold standard. If you are only studying Europeans, you will almost always see a paragraph in the discussion talking about, the downfalls of only studying Europeans and whether maybe they already are thinking about studying it on another population, or it's been done or hasn't been done. There, at least now, will always be a paragraph. And if there isn't, that's something as you students should be critical about, looking for those types of paragraphs and discussions. And then, of course, in the clinic, you'd hope that there would be some sort of validation before even reaching the clinic. But yes, as whoever's administering these drugs or whatever it ends up being should, also be very cautious of what it was originally studied on. Yeah.
Just to add on to that, wouldn't it be beneficial to have like the population in the title, like a headline, if it's going out to the general public, because anyone can just read like this SNP or tag SNP is causing this disease and think it's for them, but it could be for like a European descent, it could be for a different population.
Yeah, that's a great point as well. So we should have in the title of the paper whether it was only studied in one population. Again, that is getting better now because we are so much more aware of it. Same thing, journals will likely suggest that to be in the title or in the abstract or something right off the front where it's an obvious, so they'll still publish it, but make it obvious. And so that is something that is definitely better, and then you will see that in papers. And again, if you don't, you as students, this is a stage to be critical of that and look for that.
I was just wondering, aside from research, if they are doing some kind of skincare products that's geared toward more Caucasian kind of European descent, so would they be able to put something out there that would be allergic for Asians or Blacks?
That's a great question. I don't know. I'm not familiar with it, but just based on at least the example that I gave, if you can identify, where, sorry, I should repeat the question for those online. Is it possible to get sort of a skincare product that would be more applicable to Asian descent based on these skin sensitivities? And so, yeah, I don't know the answer, but I guess if you do understand like these variants and can test for them, then maybe there is a way to develop these sort of products to try to avoid it. Yeah, that's the best I can give. I don't know, but I imagine.
Thank you.
No problem. Any other questions? This is a great discussion. I'm really pleased with this. Okay. Going to move on. Okay, so our next thing to talk about. So this is another criticism of GWAS. So there's this hypothesis called the common disease, common variant hypothesis. And this is basically the hypothesis, as the name suggests. If there is a common disease in the population, I hypothesize that the variant that it causes the disease is also going to be common in the population. In humans, at least, this is not so true. It holds a little bit more true in plants, but still is quite a generalized hypothesis. So this is called missing heritability. All of a sudden, they actually do get to test all of these common variants and realize that in fact it does not make up 100% of the variability of a disease. So where did the rest go? And so this is our GWAS SNPs here. These are our common variants, and there are tons of other variants that can exist. Many can be, more rare variants, de novo variants, Mendelian subsets, many other that are going to contribute to the variability in disease. And so this is, again, another sort of criticism of GWAS. Again, GWAS is fantastic because it can look across the genome, don't get me wrong, but you're only looking at common variants. And so, again, something to keep in mind, that there are going to be other factors that are contributing to the disease, and you may not pick up you likely will not pick up every single one when doing a GWAS. But you will pick up those common ones because we're looking at those SNPs. Does that make sense? The missing heritability case, you might hear that a few times. Okay. So this slide I mainly just put, so you guys had it in your notes, this sort of is a nice summary of the good and bad that we had just sort of talked about. And so, yeah, it was just sort of a nice thing that I wanted in your notes, but we discussed most of these throughout the lecture. Okay. And last but not least, we have our last learning objective, which is to analyze the results of GWAS data and identify its wide breadth of applications. So I, throughout the talked a lot about humans. That's, my area of expertise, but GWAS can be done and has been done on so many different species. So let's talk about a few in plants. So I mentioned this common variant, common disease hypothesis, and I mentioned that it holds a little bit more true in plants than it does in humans. And so there's a lot of GWAS being done on these complex traits related to flowering time, growth rate, yield, and it can be really important for crop production and different plant adaptation, especially if we're looking at, let's say, climate change. There's a lot of changing environmental conditions. What plants are doing better in the changing environments versus not so good, and what is the actual genetics behind the ones that are doing better, and can we use that to increase our crops and adaptation. So one plant species that is commonly studied as a model organism is Arabidopsis. So the linkage disequilibrium is shown to decay within 10 kilobases on average, which basically, just to put it here, as the optimum number of SNPs is about 140,000. So this was a really big study done. This was published in Nature in 2010. And so it looked at Arabidopsis, and this is an important model. You might probably have heard of this model organism already, but it is important because it is widely distributed and is predominantly self-fertilizing and has lots of genetic diversity, and it has important traits for adaptation. So it's often used as a model to study these. So they had genotyped 250,000 SNPs, and they looked at 107 phenotypes, which is huge. This was a big paper. And so they categorized those phenotypes into 4 very general groupings. So the first was based on flowering under different environments. The second was related to defense, so maybe the recognition of different bacterial strains. Third was based on element concentrations. And the fourth was about developmental traits. And so the first thing that they saw was that there was a higher number of associations than they expected. So in this first plot up here, this is the bar that was the expected number of associations, and you can see that they are much, much higher than expected, especially in the flowering category. And what they realized is because there was a lot of population substructure. And so when they corrected for this substructure, they get this bottom one, which is a huge reduction. So it eliminated a lot of false positives, right? It's interesting, it showed us that there is relationships with substructure and these different traits, especially this flowering trait. But of course, it was important, as we've been talking, population substructure, is important to correct for, and we reduce these false positives. So this part B is the slide that actually shows those that they took on to study further. Okay, so this is an example that they had got. So we got our pretty Manhattan plot that we've seen. We have this one very big line. This is our significant association. And this was for the gene RPM1. And they found that it was related to a bacterial response resistance gene. And so they had identified this as an example, but they actually identified many different genes involved in different disease resistance responses based on this study. So that was one really interesting application that they found. I thought this example was also really interesting. So we've been showing these Manhattan plots as these like big peaks, but in one case they actually found, they called it more of a mountain range. So they suggested because there's such high levels of LD in this region, they didn't actually identify a single peak, and they just see this sort of big mountain range. And so this was based on flowering time in this, it was called the FRI region. And so yeah, they said it was likely explained by high linkage and population structure. And the different plots here were just them trying different corrections, and it still showed the same sort of overall mountain range. Okay, two other examples from this same study. Like I said, it was a really big study. They did a lot. This one was unexpected, they had said. So the delay of germination 1, dog 1, was not originally thought to be involved with flowering, but was highly associated with 20 different flowering phenotypes. So this was a sort of bigger result that they had found. And then the accelerated cell death 6 was they found to be associated with several lesioning and chlorosis phenotypes. And this was in fact experimentally confirmed as well. Okay, so any questions with that example before I move on to a different example? Yeah. Oh, okay, let's go up here first.
Oh, sure.
Yeah.
Okay. So like in a study like that, right? How would they know that they're not like they don't have the winner's curse or whatever?
Great question. So in a study like that, how do we know that they don't have the winner's curse? And so at this point in the study, so I think that when did they publish? 2010. So it's still pretty early days, but they did do, the multiple correction and the population substructure corrections and things like that. And so they obviously reduced a lot of that false positive that way. So I guess it still is possible, that there are false positives. Something like this one, for example, where they found that association in many similar phenotypes is another sort of type of validation. It's not common, but it is almost just to the reader validating that it's not like they just found it in one, they found it across the board in phenotypes. So it's more likely that is not going to be a false positive. But when looking at such a big study, it is possible that they still have reported false positives. Did we have a question at the back, or did we answer it?
No, I was actually just going to ask you out of curiosity, what in particular stood out to you about this, about this study? What did you particularly? Because it looks like you got quite a bit of information on it.
Yeah, so for me, it stood out how big it was. That was one thing. I think it was one of the sort of biggest papers in the field, and publishing it in Nature also kind of shows that this is a really important journal. And so that, and then also just looking at like the different, like, so this was just a few of the results that they found that, like you said, it stood out to me. The population substructure, great learning opportunity that we've been talking about how important it is. I found, especially like this one, I found interesting to be able to identify something that wasn't already assumed is a big deal, and identifying it in 20 different phenotypes. I think that's a pretty big result. Yeah, so that answers your question. Yes?
So in plants, they can make the crosses they want, not like in humans. Like they can cross the plants the way they want. What does that change for GWAS, the fact that you can actually choose the crosses you make?
Well, that's a great question. So with plants, you can cross when you can't ethically with humans, and what does that change with GWAS? So I guess you would have, and this is just me, I don't work on plants, so this is just me, I hypothesis. So I guess there would be more generations, so you might be able to maybe shuffle, make more genetic diversity. I don't know, that's, does anyone?
The thing is that they can choose the size of the algae blocks they want, right? Because you can take 1 or 20 generations, so that makes completely different sizes. So you can aim for the exact spot that you want.
Great, so the answer is you can aim for the exact spot because you can adjust the number of generations you need, which in humans, of course, we can't play around with, but in plants we can.
And also what you mentioned, like you can get rid of population structure, but just taking individuals, and so there is no like geographical variation or other type of structure.
Right, you can also reduce the population structure, so there's less of this sort of geographical substructure involved in plants as well. Okay, moving on. So another important species is maize and rice. So they have linkage disequilibrium decay is approximately 2,000 base pairs on average, so it's 5-fold shorter than Arabidopsis. This means that they need much more tag SNPs in order to look at the genome because there's less of these, shorter LD blocks. So they need about 1.6 million SNPs to be able to look at the entire genome. And so this is an example in rice. So this was published in 2011. And it looked at aluminum toxicity in rice. So aluminum toxicity is a big limitation in crop production, and this species of rice is more tolerant to it than others, and so they wanted to look at the mechanisms of this tolerance in this strain. So they looked at 383 diverse rice accessions. They took 36,000 SNPs and looked at the total root growth to the aluminum tolerance. They took, again, common SNPs, so the minor allele frequency, also referred to as MAF, greater than 0.05, which is 5% of the population. And then again, they corrected for subpopulation structure, which they actually found was highly correlated with this aluminum tolerance. So they kind of looked into the different subpopulations a little bit closer. So this is their group of Manhattan plots. So this is, they split it up by the different subpopulations because they were seeing a lot of the population-specific structures and significant hits. So they found 48 distinct regions that were related to aluminum tolerance. Only 2 SNPs were actually identified across the subpopulations. So out of that 48, there's a lot of the sort of specific population variability. And so this is just sort of 2 examples here. One of the subpopulations, indica, had 32 significant regions, and then a different subpopulation had only 1 very highly significant region. And so yeah, they really emphasize the difference in these different populations. And so they went further and took this the AUS subpopulation that only had 1 significant region. They looked at that region further and identified 11 distinct haplotypes. And so this is a haplotype analysis. This is where it's showing us the different haplotypes for all 11 combinations. And they found that haplotype 1, right here at the bottom, was unique to this AUS population, the one that had this as its sole significant, highly significant position. But they actually found that those individuals, or those strains that had this, were the strain that actually was the least tolerant to aluminum. And instead, the accessions that had highly or moderately high aluminum resistance actually had intergression, which is essentially happening from hybridization. So they essentially have a different subpopulation's haplotype in that region. So they are associating this haplotype 1 to actually a low more sensitive to this aluminum. That's my last slide with rice. Any questions before we move on to the last and final example?
Sorry, could you clarify introgression again?
Yeah, introgression. So this is where it's sort of through like hybridization. So instead of having the identical haplotype that's known in the population, you might instead have a haplotype from a different species that's based on the hybridization of the two species being combined.
I did not understand that.
Sorry?
Integration.
The integration. So let's say we're taking this region as an example. So this is the haplotype commonly found in this population. And so in a different population, they might instead have a different haplotype in that region that matches a different species, and it's come together through sort of a hybridization or a breeding of the two species. And so instead of its usual population haplotype, it'll have a different one in its replacement. In this example, it's saying that is sort of the key haplotype to the resistance of certain strains over others.
There's a question in the chat.
Oh.
You can open it.
So is this an example of outcrossing? Yes, I would think so. And when you talk about correction for subpopulation structure, is it a general statistic method or it's a case-dependent way to correct for each of our interested species? It's usually a pretty standard statistical method. So if you can identify using, let's say, like a principal component analysis is a typical way to identify it, you can see the different structure in the population, and then you can use those principal components to actually include it in your model, which will sort of correct for it. So it's more of a standard statistical practice. Any other questions? Okay. The last example I have is more of an application of GWAS. So this is pretty popular these days. It's called a polygenic risk score. This essentially takes all of your hits from GWAS and puts it into a risk score. So if you're testing for a particular disease in GWAS, it takes all of the variants you've identified, put it into a score, so theoretically you can test another individual that you didn't originally have in your population and see what is the risk of them developing that disease based on the gene or the SNPs that we're testing. So let's go into this a bit more. So up here we have our GWAS summary statistics. So for each position, so let's say we had 4 significant SNPs in our GWAS. We had the allele that is associated to it, and the effect will have a number. So whether it's a positive effect, a negative effect will be, that's what that positive versus negative. So then we have our genotyping data from a set of individuals. These can be unrelated to the GWAS data. This can be a different set of individuals. And we have their genotyping data. And so for each position, for each individual, you'll go through and add up the numbers from the GWAS. So let's say individual 1 as example. They have an A and a T at SNP 1. The A is associated to a 1.5, so we put a 1.5. Then we go to SNP 2. CG, well, the C is associated to a minus 0.5 in our GWAS, so we put that in our score. SNP 3 is 2 T's, and 1 T is a +2, so we end up adding a +4. And then finally CC, which is not in our GWAS, so it just has a 0. So this score is of 5. So then you're going to take our distribution of all of those in the population, which is shown here. The risk score is going to be on the x-axis. And so the higher the score, which would be up on this side of the axis, will be a higher risk of whatever disease you were testing. So for example, individual 1 and individual 2 have a higher risk of the disease compared to individual 3 and 4. And similar to what we've been discussing, you often will correct for age, sex, and ancestry in a similar way that I just described using the statistical methods. So let's go through, oh yeah, question?
I'm gonna ask, like, where are they pulling out these numbers from?
So this is coming, so where are the numbers coming from? This is coming from the GWAS. So in your GWAS, you'll get whether it's significant, and you'll get an effect size. So the direction of the effect, that linear model we were saying, and how strong the effect is. And so you can use that to weight your score. So if they have the same genotype, then you found it as significant in your GWAS, then you'll add it to their score. So it's sort of a summary statistic that comes from the GWAS that you can then use like a weighted score to add up from all of the different SNPs.
I have a question. So
Yeah.
And these are just SNPs that have been known that have been shown to have known associations with the disease, right?
So the question is if these are SNPs with known association to the disease. Yeah, so this from your GWAS. So these are, again, your tag SNPs. Don't necessarily know that it's the causal one, but in this case of polygenic risk scores, it's not critical that it's the causal one because we're still seeing that same linkage. So we can use our tag SNPs from our GWAS with significant associations and use that for our risk score. Yeah.
So the people that have C at SNP2, this decreases their probability that they have the disease, right? Since it's negative.
Right, so the question is if they see at SNP2, since it's a minus sign, yes, it would decrease the risk. And so if we think of our sort of linear model again, this would be the one at the lower range, for example, depending on the linear. But you know, you have 2 homozygotes, so it would be the one on the reduced phenotype range.
So it is associated with not having the disease.
Right, so it's almost as if the opposite, the other allele that was being tested is associated with having the disease. So this would be associated with a decrease of the disease. Okay, so let's do an example. So this came from 2018 from Nature Genetics. So they developed and validated a genome-wide polygenic risk score from 5 common diseases. So we had coronary artery disease, arterial fibrillation, type 2 diabetes, inflammatory bowel disease, and breast cancer. So we take our association statistics from our GWAS, and we basically from there collect those as our candidate SNPs. And then we can use a validation and testing from a new cohort to test the results of this polygenic score on these new individuals. So this is an example from the coronary artery disease. So what they found is, so this is a polygenic score on the y-axis, so a higher score is a higher risk for coronary artery disease. And they sure enough found that those individuals with the disease had a higher score than those controls. So this seems to be working. Same thing here, just a different way of looking at it. So this is the prevalence of coronary artery disease, and the x-axis is the percentile of the polygenic risk score. So again, higher percentile, higher prevalence of the disease. So it seems to be working, and this is the region of those that we would say would have a high risk, and it's just breaking it down into those percentages. So 0.5% versus the lighter blue is 2.3%, and then the lightest blue is an 8% of the population. So it seems to be working. Question? Yeah?
Yeah, what's the difference between risk and association?
Okay, so what's the difference between risk and association? So the association is coming from the GWAS. This is just saying whether you're testing the significance. If we get a significant p-value, then it is associated. The risk is coming now collecting all of those associations to get an overall risk. So just knowing one of them so the association will pick up for one SNP. Just knowing the results of what that one SNP does might not explain the whole disease, right? We're talking about the polygenic. So these risk scores are taking from every single one of those SNPs, which gives us the risk of having the disease based on if you have those SNPs or the genotypes. Is that clear? Yeah. Any other questions? Yeah.
So this is for polygenic, but for monogenic, you mentioned earlier. Is it similar, or is there a different process?
I mean, theoretically it'd be similar, but you don't actually have to add anything up because it would just be for one, right? So you can just look, does the genotype that they have match the genotype that's associated to the disease? Yes or no. This just is more complicated because you have to add up all the different sites. But for monogenic, you just have the one. Great. Any other? Okay. So let's just finish up for this example. So basically, this is an example. So polygenic risk scores show this potential for being able to identify individuals that have maybe a higher risk of a certain genetic disorder or disease. And so it opens opportunity for maybe chances of early detection or prevention of different diseases. There's often a lot of ethics discussions around this. So if we have these risk scores developed, should we test them right at birth for all of the known common diseases? Will that do more harm than good? We don't have to discuss or answer that today, but that is something that is often talked about. And then again, the same equity issue we talked about earlier. If these are being developed on primarily European participants, then it really should be validated on other ethnic groups before being used for these risk scores when it can't be generalized to many individuals. Any questions? Okay, that's it. Key points of today. So we talked about that GWAS is used to identify multiple variants associated with a complex phenotype. We talked about linkage disequilibrium and haplotypes, which are critical to investigating these SNPs genome-wide. Multiple testing and validation are crucial for reducing false positives in GWAS results. And GWAS does not include rare variants and is currently primarily explaining European populations. So that's all from me. Any final questions from this lecture? Nothing in chat, nothing in person. All right, thank you everyone.
Just if you don't have a think over, just in case there are questions, maybe there are some things about the exam or whatever things we'd like you to take over. So yeah, the GWAS session is finished, but I'll stay online in case there are other questions. Thank you very much, Michelle. How's the microphone? Is that okay? Is that working? Yes, like I was saying, for those online who could not maybe hear, I'll stay online and I'll stay in the room until the end, basically, in case you have questions. So if you have questions about the exam, about the blog post, about today's class, whatever you have, I think it's a good time now to discuss. You don't have to, the class is finished, so if you're really knocked out and you can't take it anymore, you can leave now. But since you're here to learn and since you're actually paying for that, then that'd be fair if we left just now. So there's a question, just regarding population bias. Should we sample an equal number of individuals from each population, or should we sample each population proportionally to the whole meta-population? Just a sec, I'm going to change mask because this one is way too hard to breathe, and I'm going to answer the question. Okay, so the question is about the
Should we sample equal number of individuals from each population, or should we sample each population proportionally to the whole meta population? I'm not sure there's a right answer to that question. Maybe Michelle knows better than me, but let's say the way I see it is, it also depends a little bit on what you want to do. So this bias with, let's say this clear European bias that was reported, that's an issue if you're looking for associations. But if you're looking for mechanisms, it's pretty clear that these mechanisms are biological and they're not going to be, like DNA has got completely different biochemical properties in that population or that population. So as long as you're looking for a mechanism, I think these biases are not that important, because in the end you get to the mechanism. But if you stop at the association, if you don't have the causal SNP and you're making judgment calls based on you're at risk, you're not at risk, based on things for which we have no mechanism, basically, then this is where the problems rise, I suppose. They rise most prominently. And so what would you say, Michelle, to that question?
Yeah, I think, at this point you take what you can have. So because we have a issue with how many samples we have, then it doesn't necessarily need to be equal. If you can make it equal, great, but there's no requirement for it.
So yeah, the answer is you take what you have, you make it, it depends on what you have. And there's no requirement for making it equal. Yes, indeed. Of course, like the lack of availability in some populations, I suppose in Africa, they don't really get their genomes tested every day, etc., and that makes a huge part. That explains one part of the bias, which is kind of bad in the sense that most of the diversity is in Africa, and because they've got these smaller linkage disequilibrium blocks, like the negative side is that you got to do more tests. But think about it, right? If your linkage disequilibrium block are smaller, it means that you got more chances of finding the causal SNP inside. So there's a bit of a trade-off. Like when you have large LD blocks, linkage disequilibrium blocks, you need to use fewer tag SNPs, and then you can cover the genome faster. But because each block is bigger, it's going to be harder to find the SNP that causes the disease, or that's causal, because sometimes you have more than one SNP, of course. If your blocks are small, then you need to work a bit harder to make more tests to find these associations. But once you do, you have much fewer candidates in this LD block that can be your causal SNP, right? So working with population with small LD blocks, if you're looking for causal associations, is kind of better, because you're going to be able to zoom in a little bit faster. I hope that's clear for everybody. I'm going to maybe go back to what did I do here? It's because they were. Go back to one of the slides, which I think summarizes a little bit the strategy, just to make sure that everybody gets it. So this concept of linkage disequilibrium, if you don't get it, or if you don't get it in full, it's okay, it's normal. This is by far the most complicated concept you have to learn in biology in your undergrad. Right? Let's be honest about this. This is by far the hardest. Many students at an advanced graduate level are still not able to define it, still not able to use it properly. Many professors still have a hard time with it. So if you don't get it, if you didn't succeed very well at using it at the quizzes, it's okay, it's normal. Like the reason why we're bothering you with this is because it is important, and it does take time before you can actually get familiar with it. So I think, yeah, that's the slide that I liked. Full screen again. All right, so exactly how does linkage disequilibrium play in GWAS? Where we have linkage disequilibrium, remember, it means that knowing the state of the SNP here, at that SNP, tells you the state of that SNP here, the other one, okay? You're not sure at 100%, it's just an association, but if I know you SNP here, you got this variant, then I also know your variant there, which of the two variants you have. This is the definition of linkage disequilibrium, okay? If I know something at another site, based on the knowledge of your SNP at one site, then that's linkage disequilibrium. So the older that population is, like in Africa, and the smaller are the blocks, which means that I'm learning less and less as time goes on about the other sites in the genome. Just because they recombine, the population is old and everything mixes. But when the population is young, when there hasn't been a lot of time for this mixing to happen, like one of the very famous cases, Quebec, for instance, because the population was colonized by very few settlers and grew so fast that it's actually a genetic outlier, if you want, in terms of the surrounding populations. So in that case, the linkage disequilibrium blocks are much bigger, and knowing one allele tells me actually a lot of other alleles, because these people didn't have that much genetic diversity to start with. Okay, so that's kind of the idea of linkage disequilibrium. But over time, this breaks down, and that tells me less and less about the variance at the other sites than the one I'm looking at.
Is there linkage equilibrium? Like nothing.
Is there linkage equilibrium? Yeah, theoretically for sure. Like if you wait long enough, literally every site will be broken by recombination, and even if they're really close, I will learn nothing from the site nearby, because there has been a lot of time for breaking these recombinations. So all the possibilities are in the population. Right, so that's the idea. It takes more time to break these associations when the sites are really close, but given enough time, that will happen. In practice, that never happens though. Okay, like in theory, if you wait a zillion years, then that would happen, but in practice, that never happens. Also because you have the creation of new mutations. Okay, and that kind of creates new linkage disequilibrium that are just appearing because you get these new variants. But in some places you can get reasonably close, maybe not in the human population, because there was a very rapid expansion of the human population, and let's say, of course for us it seems like a long time, 200,000 years, something like this, but when you compare it to other species like plants and especially fungi, you know, like they have been around for much longer with bigger populations, and then you will have much more small blocks of LD. In humans, we will always have these LD blocks that are taking at least a couple of SNPs in a row, that's for sure. But theoretically, they could be much smaller if you wait long enough. Okay, so that's where LD plays in. You assay that SNP, and you don't need to assay this one because you know it already. Okay, it's within the block, and the block is fuzzy. You know, there's not a proper beginning and end, but you say within this XKB or X megabase. Like if I know this one, I also know this one. So I can pick this one as my tag SNP. I could also pick this one, right? I could pick any of them, because once I know one, I know them all. That's the idea of these LD blocks. And actually, that's what makes it easy, but that's also what makes it hard, in the sense that if the LD blocks are big, then at the time when I want to know the causal one, it's going to be a problem, because I won't be able to tell which is the one that's the cause. There could be 1 of 100 if the LD block is big, or 1 of 10 if the LD block is small. So that's really how this notion of linkage disequilibrium plays in GWAS. Okay, and if you still don't get it, if it's still hard, it's okay, it's normal. That's a very hard concept. It takes years and years before you get comfortable with it. Right, Michel? It takes years. So that's the that's the idea of how LD plays, and why that was important to introduce LD before we see GWAS. Other things you'd like to discuss? I mean, it can be about this class, can be about anything. How is this going for you guys with the blog post reading? Have you started to look at the whodunit, etc.? How is that going overall?
Yeah, I actually have a question. So on Quarkus, the final version of the blog post is on 21st of April.
Yeah.
And on the registrar website, it's like the final exam is like due on 26th.
So the register says 26.
Yeah.
The registrar is always right. Even when the registrar doesn't tell me anything, the registrar is right. So I'll change the deadline. I have pushed so that it would be as far as possible in the semester. So the advantage is that it gives you more time to write the blog post. That's an obvious advantage. The disadvantage is that some of you are waiting for their grades for graduate applications, etc. So there's a bit of a trade-off, and I've chosen to push it to the maximum so that you guys have as much time as possible to write a good blog post. I know that the end of this semester is pretty busy, and you guys will be kind of, also with other courses, other deadlines, etc. So I hope it's okay with you that I've pushed that to the maximum. And the registrar is pushing the other side. They tell me, no, like we want the grades as early as possible because of the graduate applications that you guys have for some of you. So we both push, and then the registry tells me the maximum time. But after this, they change it. That's why there was a mismatch. Okay, so the idea is I want to give you as much time as possible to write the blog post. After this, they give me 1 week. They say at the time when the final exam is given, I have exactly 7 days to give them the final grade, like close everything, lock it, and say these are the final grades. They cannot change anymore, even if people complain. So that's also the constraint on my side, is whenever the deadline has come, then I have only 7 days to do everything, which means that you can't really be late for the blog post. Okay, you can have your joker; it's gonna work, no problem, as usual. The rule is gonna work, but it's going to cause some additional difficulties. And one thing that I haven't talked about much yet is the fact that after you submit your blog post, there is one last activity, which is kind of fun and doesn't take a lot of time, is to read other people's blog posts. Okay, so it's really nice to know one article very well enough so that you can explain the story in all detail. But it's also nice to know what other students have done. And the way it's gonna work is that you can optionally decide to put your blog post for comments in the pool, and you're going to receive criticism from other students. And for every time you give criticism to someone else, you're going to get marks. And even if you get good criticism or bad criticism, it doesn't matter. You're going to get the same amount of marks. Anyway, the criticism are going to be structured. I'm not going to let you just say whatever you want. There's going to be kind of a rubric. And so either you're going to be part of that feedback session. In that case, you will expose yourself because you have to show your work to other people, and that's always difficult to hear what they have to say when this is negative, and there's always things to improve. So you will have to expose yourself. The benefit is that you will be also able to help other people improve their blog post, and that will give you bonus points. If you're late for your final blog post, then you won't be able to participate to that feedback session. Okay, so you won't be penalized as long as you got jokers, right? You can use your jokers for that, but you will be penalized for the points you could have made by participating to that feedback session. I'm going to say more about this next time, more concretely, but just don't plan on submitting your blog post late. That's my advice, okay? Plan on submitting on time, because they will give you access to that option to receive feedback and to give feedback. And you'll see it's actually quite fun to see what other people have written. It's always a good amount of fun. Yeah.
Professor, will the criticism we get affect the grading?
No, good question. Will the criticism we get affect the grading? No, of course not. And that is forbidden. Why is that? Because otherwise we'd have to pay you because you'd be considered teachers. So, like, there had been mishaps in previous years that, there was some kind of grading that was based on feedback from other students, and they had to pay TA hours to the whole class. And so, like, we're really strictly forbidden to do that. So I'm deciding what the grade is, and independently of that, there will be feedback that is given by the other students. So that is useful. That is not useful for the grade as recipient of the feedback, but that is useful if you want to publish your post online, because other students are your typical reader. So if they say this is hard to understand, it means that it's going to be hard to understand for your average reader online. But no, so I insist, this is absolutely not going to impact the grades. This is totally forbidden by U of T. Other questions? So have you started to look at the whodunit or not yet? Still have time, of course, and remember that the deadline is even after the last class. So you'll see about this. It's not really easy. Also, the difficulty with the whodunit is that by now there is substantial amount of sharing between people, which is, that's what I like. I want you to learn from each other. But too much sharing kills the sharing a little bit, in the sense that giving the answers to other people is not what I hope would happen. I would hope that people would just brainstorm together and then help each other find the right answer. But it's kind of, the purpose is not to make it easy for people who come last minute, say, oh, what's the answer again? Oh yeah, grabbing it from the website, putting it there, and then I work 3 minutes when everybody else worked 3 hours, getting the same grade. That's not at all the idea. So it was kind of hard for me to find examples that would be somewhere in between, where you could exchange information with each other without really telling away the the answer of the problem. My advice is don't start too late, because I had to increase a little bit the difficulty because of the amount of sharing I see, that I infer, let's say. I don't see the sharing at all, but that I infer from all the indications I have from the quizzes. You laughing? You find it funny? Yes, what's your question?
So, you know how Kyle's blog post
Yeah.
is a good example?
Is
Like-
Kyle's blog post a good example? It's a standard example. I mean, it's neither particularly good nor particularly bad, I think. You will see that Kyle's blog post had, I think, 4 figures, if I remember, when I'm asking only 2. But more figures is kind of always better. If you feel like it, then you can make more figures. But he overdid it in terms of figures. And otherwise, in terms of text, it was average.
Is it possible to get like high 90 and above on the blog post?
Can you get 90 and above on the blog post? Yeah, it's possible. Some people had max grade last year. So I'm not a particularly evil grader. I'm not a particularly, especially Michelle, because she's been doing the grading on the midterm, and the grading on the quizzes is completely automatic. So I'm not a completely evil grader, but I'm also not a particularly nice one. When something is, as good as can be expected from an undergrad, I don't hesitate to give max grade. Still, it's kind of it's work. I can't remember the grade of Kyle, but it was not max grade, if I remember. His second blog post, the one that he made outside BIOD25, is much better, in the sense that he also had much more time. It's exactly the topic he wanted to develop, etc. So my advice would be look at his second blog post. If you have the time, if you have the the dedication to do what he did for the second blog post on your first blog post, that would be a very, very good example of how to maximize your grade with the writing. So one thing that's quite important for the blog post, let's say that there's what's in the rubric, because I have to have a grade to say you pay points for this or for that. But still, one thing that's really important is that your text is faithful to the article. Well, it's as simple as that. But if there's a big mismatch between your text and the article, then I won't be able to give a lot of points. Like, you still have to stick to what is in the article. So make sure that you understand the article enough at that stage so that you will be able to describe it in written form. Right? You don't have to write anything now, but my advice is make sure you understand the article. Some of the examples I saw with the titles tell me that some people haven't really gotten the gist of the article, which is not a very good start when already the title is a little bit off. So make sure you understand the article. That will play a huge part in your grades. The other thing that's quite important is just the logical consistency and the presence of mistakes. If you're telling me that humans have got 109 chromosomes, for example, I will have to penalize or things like this, as you understand. So just make sure that whatever you say is factually correct. Make sure that this is logically connected with your narrative and that this sticks to the article. That's the most important by far. 2 minutes, we still have time for plenty of information.
For the title, I don't think the marks are out yet, right?
No, the marks are not
I was
out yet for the
wondering if
title.
you give like individual feedback
Yeah.
for the title.
There will be individual feedback. You're not going to get just a grade. You're going to get like, okay, this was improvable.
I think in terms of description for the article, like making that balance between that it's not, your blog post is not just a summary of the article, and you also have introduced some new ideas.
Yeah.
Is that something that even if our TLDR did like reflect that, oh, this is just a summary of what I've read, but this is a new idea based of what has been introduced in that article? Like, how do you make that balance? Because there could be a new idea that isn't 100% yes, this is what the article is saying, but it's drawing from some of the ideas that have been posed in the article.
Yeah. So the question is how do I make the balance between the new ideas that you're supposed to introduce, the ones that are in the article, and the TLDR. So the TLDR, for now, I'm not going to assume anything about what you're going to write later. It's just to make sure that you're making progress and understanding your article. At the time when you write the final blog post, most of the new ideas and the things that you're going to introduce are actually in the introduction. Okay, because you need to give the background that is necessary to understand the article. An academic article assumes that people have at least a certain level, usually a PhD, or let's say in the process of acquiring their PhD, to be able to understand. You cannot make these assumptions for your readers, so you have to kind of give the background that they need to be able to understand the conclusion. So most of the ideas, to be very blunt, are going to be in the introduction, the new ideas that you're going to have to introduce. Okay. So that's kind of a background check. It's as simple as that. You don't have to reinterpret the article and give different conclusions than the ones of the authors. Okay, it's time. So thank you very much for your attention, and see you next week.